Good morning, good afternoon, or good evening, wherever you are. Welcome to our Expert Insights webinar series. We are waiting for a few more guests to join us, so please be patient. So welcome to those joining now. We'll start in 2 minutes. We'll start the webinar. Okay, so good morning, good afternoon, and good evening again, wherever you are, whatever the weather is. I'm based in Belgium, and we have snow at the very moment. I do hope you have a better weather on your side. My name is Martine. I am the Director of Professional Education at CooperSurgical Fertility Solutions, and I'm a member of the Professional Education and Clinical Support team. I will be your host and moderator in this webinar today. So welcome to our new and exciting new webinar series, Expert Insights, where we will invite peer-to-peer experience sharing, and we will focus on cutting-edge expertise. This series, we designed it so that it is a platform for professionals to share their insights and experiences on methods and processes in the IVF lab and clinics. So thank you for joining us. And we will bring together different key opinion leaders throughout the series. From both the lab and the clinic, we will discuss new methods that are shaping the future of our field. We have received many questions beforehand via your registration, but please continue to submit your questions during the webinar to our experts, and you can use the Q&A button on your screen. We will address those questions during the Q&A session at the end of the webinar. This webinar will be recorded and will be available for download on demand, and it will be posted on our CooperSurgical Knowledge Hub website soon. Today's episode will focus on andrology procedures in the ART lab, more particularly on sperm prep, including a sperm separation step, and by using the ZyMōt device, and this could be for IUI, IVF, or ICSI procedures. Oskar and colleagues described already in 2014 that an ideal sperm separation technique should accommodate large volumes, have high and efficient enrichment for motile sperm, isolate sperm with higher velocity, isolate sperm with normal morphology and nuclear maturity, reduce Ross generation, reduce morphological damage, reduce the percentage of sperm that have DNA fragmentation, and it should be rapid, it should be cost-effective, and labor not intense. So this is what we're here for today. We will explore the scientific data and outcomes, as well as the practical aspects like validation of the method, the training in the lab, the laboratory efficiency when using the ZyMōt sperm separation device. We have invited two peers, Neelke de Munck and Emma Adolfsson, to share their experiences with us. Please do note that the experts are expressing their personal opinions and perspectives. So first we will meet with Neelke de Munck. She is the lab director at the Brussels IVF at the Free University of Brussels in Belgium. She finished her PhD in 2010 when she focused on oocyte putrefaction. And in 2018, she moved to the EVRMA ART clinics in Abu Dhabi. And then she came back to her home base in 2021, being the lead director of the IVF lab at Brussels IVF. Her main focus is optimizing efficiency in the lab, but also increasing the quality and the safety for IVF procedures offered to patients. She will be discussing her work and her team's work, of course, on the ZyMōt sperm separation device that was published in the Journal of Assisted Reproduction and Genetics. Our second speaker is Emma Adolfsson. She is from the Örebro University Hospital in Sweden. Emma is working on her PhD, and she's currently a team member and she of the IVF lab there, but she's also doing her PhD in a different domain. So she's doing a lot of research, and that is in the precision diagnostics, exploring the possibilities of next generation sequencing. She teaches a lot, she supervises, but she loves working in the IVF lab. She has published a lot and she lectured a lot already, so I'm really looking forward to her contribution today. She will elaborate on their study on the clinical validation and experiences of the ZyMōt sperm separation device. She will also demonstrate some clinical data. This study is now accepted for publication in RBM Online. So I'll give you a short introduction on the ZyMōt sperm separation device. It is for sperm separation. There are two types, the 850 microliter device and the 3 ml device. It separates sperms based on their motility. And I have a little video for you showing how that really works. So this is the device. It has an inlet and an outlet. There is an inner chamber and an outer chamber. And the sperm, the ejaculate is entered or flushed into the lower chamber with a syringe. The sperm will then swim out into the device and then swim through a membrane. This is a polycarbonate membrane which has pores. These pores are 8 micrometers of size. And then the motile sperm can be harvested from the upper chamber. The sperm will accumulate there. Here is a short overview of the comparison between density gradient centrifugation and the ZyMōt procedure. As you can see, it is simpler. We don't need that much equipment compared to the density gradient centrifugation, and there are less steps involved. Does it work? This is a very recent publication from the group of Banti, and they showed that there is a significant increase in blastocyst formation, in utilization rate of a blastocyst, but also in euploidy rate of those embryos. This is a retrospective study of patients who had a first cycle 1 year before. So it is a smaller group of data, but it is important data. Fertilization rates were similar with the density gradient group. And focusing on that euploidy part, multiple studies have focused on this using the sperm separation device. And as you can see, the ones with the asterisks, those are the ones that have significant improvement, significant more euploid embryos after using the ZyMōt sperm separation device. The others have similar or a trend to better outcomes on euploidy. But how does it work? What does it mean practically for you in the lab? What are the benefits? What are the outcomes? And for which patients can we use it? I'm now handing over to Neelke to elaborate on her study. So Neelke, the floor is yours. If you can share your screen and
Yes. Hi, good morning, good afternoon. Thank you, Martine, for the kind introduction. So I will now share the experiences we have here at Brussels IVF. So just to introduce you to our clinic, so we are Brussels IVF. We are located in the center, in the heart of Belgium. So we are located in Brussels. To guide you a little bit on the number of cycles that we do, so we perform around 5,700, 5,800 cycles a year. So these are pickup cycles only. Most of them are treated with fresh ejaculated sperm, but we also use donor sperm and other types of sperm. When we look at the number of embryo transfers, I just present here for the first 6 months, and we perform on average 20 transfers a day for all those months. And then when we look at the number of inseminations, so you see in orange the number of inseminations with donor sperm, in blue the number of inseminations with husband sperm. So you see that these are the numbers that we perform in our lab. So this is a very short summary of who we are. So then let's jump immediately into the ZyMōt device. So Martine already nicely explained that there are 2 different types of devices. So you can clearly see the inlet. of both devices, the 850 and the 3 ml. Then the sperm will pass through the membrane and will reach the outlet where the sperm, after separation, will be collected. So this is what the chamber looks like. You have the inlet, the sperm will move, it will be separated, and then it can be collected. So the volumes that you can add to the device and capture from the device are 850 and 3 ml, and then you can take finally 500 microliters or 1 to 1.5 milliliters in the 3 ml device. So to have a closer look at how the separation happens, Martine already showed this in a video. I'll show you in pictures. So you insert your ejaculate, 850 microliters in this system, and you will see that the sperm will start to move to the sperm separation. And this is indeed developed from micropores around 8 micrometers. And then the sperm will move through those pores, and in the purple space you will see you can aspirate the separated sperm. So usually this process takes 30 minutes. You inject, you wait for 30 minutes at 37 degrees, and then you can aspirate the separated sperm. So what do we know already about this device? There's already quite a lot known on the device. So I will try to guide you through it a little bit and then give you some experience from Brussels IVF. So this is a study in which they looked at the reactive oxygen species. So they compared washed sperm with swim and then 3 different types of pores from a ZyMōt device. And as you can see, the generation of ROS is significantly reduced when the micropore devices are being applied. And also when we look at the DNA fragmentation, we can see that for the three different types of devices of the ZyMōt, we see a reduced DNA fragmentation. This reduced DNA fragmentation was also observed in this case for the swim up. So they conclude reduced reactive oxygen species and also reduced DNA fragmentation. Then another study has been performed in which they compared the ZyMōt device to a swim up. And they specifically looked, in this case, to blastocyst development and to the sperm DNA fragmentation index. And what you can see is that there is a significantly lower DNA fragmentation index in the ZyMōt group compared to the swim-up group. So this is in contrast to the previous study where equal sperm DNA fragmentation index was observed. And this study shows significantly reduced for ZyMōt. Then they also saw that there was a slight increase in blastocyst formation, though it was not significant for the ZyMōt group. Then when we look at the sperm preparation compared to sperm separation compared to the density gradient. So in this trial, they looked at fertilization, embryo quality on day 3, and the high quality blastocysts. And you can see that specifically for the high quality blastocysts, in this case, there was no difference between both techniques. And then when we compare the control, so the density gradient with the microfluidics processing with regards to the clinical outcomes, we also see that in this specific trial, no differences were observed in ongoing pregnancy rates and clinical pregnancy rates. So similar cleavage, blastocyst stage, embryo quality, and clinical and ongoing pregnancies when comparing DGC to the ZyMōt. Then also very interesting and also already highlighted by Martine is the ploidy outcome. So this is of course also interesting based on the specific separation of the sperm that you make. Is this also able to increase the ploidy outcome? And what you can nicely see in this study is that there was indeed an increased blastocyst formation and also an increased percentage of euploid blastocysts available after biopsy. Then, when we look in general to the sperm preparation, when we compare ZyMōt to a density gradient, you will see nicely in this graph that the total motile sperm count after gradient might be higher, but this is also because of the way the selection happens and how the concentration of the sperm will be affected by the type of technique. So this is what you can see in this graph, that the ZyMōt device is able to generate a high sperm motility after the preparation. You see 96% compared to only 84% in the gradient device. And this can give a slight difference in the total motile sperm count. But what is nice for the ZyMot device is, of course, that you have a very pure population of motile sperm. And specifically in this study, they used it for insemination. In this study, you can see each time non-significant values were obtained, but still there is a small difference that maybe refers to a clinical difference. So you see pregnancy rates of 18% for the ZyMot device and a little bit reduced, though not significant, after density gradient centrifugation. So good outcomes with IY as well. Then to indulge you with the Brussels IVF experience, so we performed first an initial trial in which we wanted to compare a washed sample, a sample after density gradient, after MAX and after the microfluidics sperm sorting, and we looked at the motility, morphology, acrosome index, DNA fragmentation, and the vitality. So these are the sample characteristics. In total, 52 samples were included. You see the age of the patients, the abstinence, and then you see that the samples were quite good, a concentration of almost 60, and also the progressive motility of almost 60%. So when we look then at the sample, what it looked like, depending on the different techniques that were used, you can see that what is extremely important is the progressive motility, where you can see that the highest progressive motility is obtained for the sperm separation device. And when we focus on the DNA fragmentation, you also see that the lowest DNA fragmentation was obtained for the sperm separation device. We then also looked at the apoptotic cells and the dead cells, specifically comparing the ZyMot device to the MAX device, because MAX should be able to deselect against these cells. And what you see here is, in fact, that the sperm separation device was even better compared to the MAX in selecting against those apoptotic cells. So because the results were so nice, we decided to perform a sibling oocyte study with, of course, primary endpoint utilization rate, comparing ZyMot device with the density gradient centrifugation. So in total, 253 oocytes were required in each arm. We included ejaculated sperm and patients with more than 6 mature oocytes to have at least 3 oocytes in each group. So you see here the general characteristics. So females were 34 years old, partner 37. And then you see after preparation, the sperm concentration and the progressive motility, which of course was significantly higher in the ZyMot device because it's able to separate the motile sperm cells. Then we also looked at fertilization, the day 3 development, day 5 development, and our primary endpoint utilization rate. And you can see that there's no significant difference between density gradient and the ZyMot device. So this, of course, enables us to try to implement this in the clinic and reduce this time-consuming nature of the density gradient centrifugation. So what do we win? If we look, when we compare density gradient with the ZyMot device, we see that when we look at our eyewitness timings, we see that the timings are almost exactly the same. We have 39.56 minutes and 38.35 minutes. The only difference is that, of course, in a density gradient centrifugation, there are many more steps involved compared to the ZyMot, and I'll come back to it later. To give you a little bit an idea on recovery rates, because this is also important for you when we look at the raw sample and how it comes out after ZyMot, I'll give you 4 examples from us in the clinic, a bit lower concentration and lower motility, and you can see that the recovery of the concentration 0.4 and 98% motility after preparation is really nice. And then as the concentration and the motility increases, you see that also the concentration after ZyMot is gradually going up, more than 1 million and 97% motility. Sample 3 starting with 28 million and ending up with 3.6 and very high motility. And then when you see very high samples, they come out extremely nice. So how do we start this in the lab? We look at the benefits. We show that it shows an improved motility. There is an improved DNA fragmentation, as in DNA fragmentation is lower. And this improved sperm preparation time gives us flexibility. So this is what I mentioned before. So even though the time for density gradient and ZyMot takes around the same time, almost 40 minutes. We see that for density gradient, there are multiple steps involved. You do your sperm analysis, you make the gradient, you centrifuge, you wash, you do your final sperm count. And this is reduced for ZyMōt. You see, you do your sperm analysis, you incubate 30 minutes, and you have your final sperm count. And this could enable you to apply multiple samples within the same preparation time, which is impossible for density gradient centrifugation. So how did we start in the lab? When we analyzed all the samples that had been done, we decided to go ahead with patients with more than 1 million per ml in the raw sample and a motility more than 10% A plus B. Frozen samples and IUI, we are still validating at this time. So how can you integrate it? You need to arrange a schedule for your sperm preparation, because if all patients only produce a sample every 30 minutes, you will not be able to treat multiple samples at the same time. So you need to ensure that the samples are scheduled in such a way that they allow you to treat multiple samples at the same time. Very important in your lab setup is that you have a division in time and space, provide an incubator in which you can do it, make sure you have a protocol available, and also train your people. For this training, CooperSurgical also provides an online training session which you can follow before you start implementing it. And then, of course, evaluate. When you start implementing it, after 1 month, after 2 months, evaluate how your KPIs are and see if you need to make any adjustments. So here on the right, you can also see the incubator that we use in the lab to put the different samples, and there can only be 1 sample in each space in the incubator. So we can treat 6 samples at the same time. And very important, do not forget to validate before you implement and evaluate after you have implemented. Thank you.
Thank you so much, Neelke. That's super interesting, especially how you implemented, how you validated everything. So now we move over to our second speaker, Emma Adolfsson. So Emma, welcome again. And please keep submitting those questions. We already have some great questions for our speakers. So Emma, the floor is yours.
Thank you. Can you see my screen?
It's not in presenter mode yet.
Is it in presenter mode now?
Now it is. Perfect.
Thank you. Thank you for the introduction, and thank you for letting me be in this webinar. My name is Emma Adolfsson. I'm a senior embryologist and a PhD student, and I work in Sweden, which is up in the north, and we do also have snow right here. And today I'd like to share our experiences of using the ZyMOt for standard IVF, because I know it's interesting for a lot of people. Can you use ZyMOt for standard IVF? And I hope to answer that today. This is my disclaimer. And as mentioned by Martine, sperm preparation has several purposes in assisted reproduction, and in the ESHRE guideline from 2015, they state that sperm separation or preparation aims to eliminate seminal plasma, debris, and contaminants from the ejaculated fluid. It also aims to concentrate the progressively motile sperms found in the ejaculate and to select against the abnormal sperm. And the guideline says that the swim-up technique and the discontinuous density gradient centrifugation are the most frequently used and accepted. However, this guideline now has almost 10 years of age. And our clinic, which is quite small compared to Neelke's, is a publicly funded clinic. We do about 500 oocyte retrievals with or without fresh transfer. We do 300 frozen embryo transfer, but we also run a donor oocyte bank, a donor sperm bank, and we provide fertility preservation. And we've been relying on the density gradient centrifugation using media from Nijs, the pure sperm solutions, for all types of sperm preps. However, we became interested in changing the sperm prep method in the clinic for several reasons, but one of them, the major one, was the increasing evidence that centrifugation compromises sperm integrity. So we wanted to see if we could find something which would avoid centrifugation. We also were interested in methods that would provide less handling, less manual handling, to remove room for mix-up errors. If possible, find something which was faster, something that was standardized, and something where we could possibly save money and or reduce waste. So we scanned the market and we came across the ZyMOt sperm selection device, which on paper appeared to tick all our boxes. So This has been presented by Martine. For us, the main thing was the number of publications indicating that with ZyMOt, you get a high fraction of motile sperm and you can get reduced DNA fragmentation index. So we move on into how we worked instead. We wanted to introduce the ZyMOt into the clinic, and we wanted to do it stepwise. So we wanted to do the validation and the introduction in several steps. So we did a 3-step process where we started with split semen comparisons, where we familiarized ourselves with the device, we trained, we compared against the current protocol, which was density gradient centrifugation, and we wanted to ensure that the device would fit within the clinic in terms of logistics, workflow, labeling, etc. After that, we moved into a sibling oocyte study for conventional IVF. And then finally, we introduced the ZyMOt into the clinic for ICSI and IVF, but I'll talk mainly about IVF today. And as Neelke was saying, we were running KPI comparisons throughout this period. So let's look at those steps one by one. In the first step, we did split semen preparations, where we used surplus samples from men doing sperm analysis in the clinic, and we selected 25 of those who had a volume above 3 milliliter. We then prepped the sperm using both density gradient centrifugation and the small ZyMOt chamber. And as you can see there in B, ZyMOt consistently gave us very pure fractions of progressive motile sperm, where about 98% of the sperm were with high viscosity and just a few immotile sperm, and with very little variation, compared to density gradient centrifugation, which is shown in C, where there were fewer progressive motile sperm with more variation. As mentioned, however, ZyMOt will give you more sperm, so it will give you a higher total number of sperm count. And in each and every sperm sample that we looked at, there was always a higher and purer number of progressive sperm after preparation. So we were happy with this, and we moved into a sibling oocyte study for standard IVF. And because we are a small clinic, we couldn't do so many because it would take a lot of time. But we had an inclusion criteria that we wanted more than 10 oocytes after retrieval. We wanted patients with normal semen parameters, and they needed to have more than 3 ml semen volume so that we could split the semen. And it takes a lot of time to do a sibling oocyte study, but we did it anyway. So the semen was split into 2 groups, and we prepped half the volume with density gradient centrifugation, and we used a smaller chamber for the other part. And then we fertilized half of the oocytes with a density gradient prepared sperm, and half of the oocytes with the sperms from the ZyMOt. And as you can see, we didn't have any significant differences in terms of fertilization rate. They were identical. The semen parameters again show that the ZyMOt resulted in consistently high, pure fractions of progressive sperm, around 97%, compared to density gradient centrifugation, where we have 88% progressive motile sperm. Again, the number of sperm were higher with density gradient centrifugation. When we looked at embryo development parameters, there were no differences. We achieved the same utility rate. The embryos were scored likewise in KID score and IDA score, which were obtained from Vitrolife. And we also checked morphokinetics. So that is the division speed, the division pattern, and they were also identical. And we were very happy with the ZyMOt. So we decided to move into the 3rd step, which is to introduce the ZyMOt as the primary sperm selection device for IVF in the clinic. Now, this happened in 2023, and we did 2 months of standard IVF with ZyMOt without centrifugation. And in that time period, we included 347 oocytes. And we noticed when we were following the KPIs that the fertilization rate dropped below what we were happy with. We also noted that the utility rate dropped, which is then the number of vitrified or transferred embryos. But above all, we noted that we had a higher percentage where we had complete failure of fertilization in standard IVF. The total fertilization failure rate increased to above 12%, which is a red flag for us. So we then reached out. Oh, sorry, I'll go through this first. To check against our normal KPIs, we pulled the KPIs from historical data, and we checked the numbers from 2017 and onwards. And in that time period, we had fertilized over 8,000 oocytes with a fertilization rate of 66%. And in that time period, in 5 years, we had a total fertilization rate of 6%, which is within the scope of what ESHRE and ASRM are stating is acceptable. And that's normal for us, given the standard procedure that we have. Compared to that, when using ZyMOt without centrifugation, we had a double risk of getting a total fertilization failure. So we reached out at the company who was selling ZyMOt at the time. And unfortunately, we were then informed that they had released a new product insert for standard IVF, saying that if you are running standard IVF with ZyMOt, you would then add 3 ml of media. After retrieving the motile sperm, you would centrifuge for 5 minutes, remove the supernatant, and resuspend the motile sperm in fresh media. And the reason for this was to remove seminal plasma factors that could otherwise be retrieved with the motile sperm and would make fertilization harder. So we were so happy with the ZyMOt as a device in terms of logistics, etc., and I'll get back to that. So we decided to continue with ZyMOt for standard IVF, but with adding 5-minute centrifugation as recommended, and now being the standard operating procedure for ZyMOt. And after doing that, the fertilization rate has gone up. So it's now in the same region as we have for density gradient centrifugation, slightly higher. And the total fertilization rate has been reduced to what is normal for us. And we've been running it since the revision and up until now. And so far, we've done more than 2,000 oocytes. So how do we in Örebro work with ZyMOt now? We use ZyMOt for a lot of things. For ICSI samples with some male factor, but not the worst cases, we will select the smaller chamber, the 0.85 ml chamber, and we do not do any centrifugation at all. We use them straight as they are retrieved from the ZyMOt. For standard IVF, we would select the larger chamber, the 3 ml chamber, in order to get as many sperms as possible. We will then add 3 milliliter media and centrifuge for 5 minutes before resuspension and doing the final sperm count. If we have really poor samples, ICSI, we wouldn't do ZyMOt, because if they don't have motile sperm, the concept of microfluidics won't work. So we would then do a direct centrifugation or possibly a 40% density gradient followed by washes. And in our clinic, we actually prep the semen prior to cryopreservation instead of preserving the ejaculated semen and then prep them. And for cryopreservation, we would then run density gradient centrifugation in order to get as many sperms as possible for the future. So in terms of scientific outcomes, the main reason for shifting sperm preparation method was to avoid centrifugation. And we conclude that sperm preparation with microfluidics and the ZyMOt sperm selection device is suitable for standard IVF, but you have to add the 5-minute centrifugation step. And therefore, centrifugation cannot be completely avoided, but we reduce the centrifugation time from 40 minutes to 5 minutes. And for ICSI samples, you do not need to do centrifugation at all. We wanted to standardize the procedure, and we are consistently seeing pure sperm preparation with a very high fraction of progressive motile sperm. We also had some non-scientific but equally important goals that we wanted to achieve. We wanted something with less handling and less room for mixed up, and we think that the ZyMOt fulfills this criteria because there are less transfer steps. You go from cup to chamber to final tube. The chamber is compatible with labeling, so we find it safe for use. Is it faster? My personal reflection is yes, the hands-on time is less. The actual processing time is the same, but you can do other stuff in between. Does it save money? Depending on how you use your staff, yes, because we are expensive. We should be expensive at least. So if you can save on the hands-on time and if you can do more in the same time, you will save money. Does it reduce waste? yes, for us, we are reducing the number of centrifugation tubes, the number of pipettes, the number of serological pipettes. We are, though, using more syringes for the use of ZyMot. You need to have a lot of syringes. So to conclude and summarize, we are keeping ZyMot as the first choice for sperm prep in our clinic. And with that, I'd like to thank you for your attention. If you want to communicate with me, you can email me. And these are my co-workers who helped me with the ZyMot work. Thank you so much.
Thank you so much, Emma. Really, really interesting and nicely explained. So Neelke, if you could join us for the Q&A. We received already many, many questions during the registration process, as I explained in the beginning of the webinar. And also now, our participants, and we have a big group of participants, already submitted many questions. So the first one is really practical. What type of incubator do you use? And how do you transport the device? Because we saw the device, but I do know what you explain how you do that. Maybe Neelke, you can start with the type of incubator.
Yeah, so we use, especially for an ICSI sample, you do not need to use an incubator with CO2. So you can just use like the Heratherm incubator, for instance, just at 37 degrees. A regular heated incubator that gives you 37 degrees. That's in fact all you need. So it's very easy equipment.
So your media that you use during the separation is a HEPAS and/or MOPS buffered media.
Yes, correct. Yeah. So this is just the HEPAS buffered medium. And then depending, of course, as Emma explained, in case you do it for an IVF, you might as well do your separation in a Heratherm and then do your final wash in a fertilization medium, because that you can do separate. So no need for expensive equipment in this case.
But you will put the device on a larger Petri dish. So because that was a question that was in the chat.
Yes. Yeah, I did not show a picture of it. This is how we do it. We have indeed a round dish in which we put the separation device, and then we put it for us because we separate the incubator in 6 chambers, each with a timer on top of it as well. So you put it inside, you close it, and you switch on your timer.
Yeah. So then, Emma, for the question regarding the labeling and the witnessing, so there is a space on this device that you can easily write or add a tag for the witness.
Yes, we use a labeling system from the laboratory information system, which is a plastic label. We don't have a great eyewitness system, so it's not an RFID tag. It's a label with names, etc., unique identifiers, and for us the labeling works really well in terms of clarity, in terms of placement, but that has to be checked for each and every laboratory, of course.
Yeah. So for you, Neelke, you will use the eyewitness tags, for example.
Yeah. Indeed, I wanted to comment on it. We use the eyewitness, the RFID tags, and I mean they fit on the device. We never had issues with it. Yeah.
So then the big question is, again, super practical but very important. How do you go about volume of ejaculate? So what is your minimal volume and the maximum? So what if you have a 12 mil, which is exceptional and is also an indication for something else potentially? Well, how do you deal with that? Will you use an 850 microliter device, the 3 mil?
I can answer, or we can answer both.
Yeah. Oh, fine.
I think it depends on the FERT method that you're going to select. If it is an ICSI sample and he ejaculates 12 mil, it really doesn't matter, does it? Because you only need as many sperms as you have oocytes. So if it's an ICSI, you're going to get enough sperm with a small chamber. If it's an IVF and the volume is very high, but the concentration is very low, then either you go for the large chamber or you even take 2 of them or something like that and combine them in the end. So you really need to start with what's the purpose of the sperm preparation? Is it IVF? Is it ICSI? Is it cryopreservation? And then you make an informed decision. That's what I would do.
Yeah,
What do you say?
I will maybe comment a little bit more on you, Emma, with our own experience that we have. And this is also what I showed in my presentation, a few samples, what they looked like in the raw sample, and then how do they come out. And you see that even in a sample with a concentration less than 1 million at start, you see that the concentration afterwards was still 0.4. And the advantage, of course, of the ZyMot device is then that your motility is 97, 98%. So this is perfectly fine, irrespective of the volume that you apply. And this was also a question that was related to this. What if your volume is lower? Because specifically for these, it might be important. So the guidelines of the device are that you can just add HEPAs to come to your final volume of 850. I did not show examples when the volume was low, but also in these cases when we need to add HEPAs to it, the final concentration is also okay. Of course, only experience with the ICSI and the 850 device here.
So in conclusion, you will or use a part of the ejaculate, or you will reduce by a spin that is necessary, or you will add some medium to get the minimum volume to be added to the device. Great. What about viscous samples?
Yes, I have sorry.
Yeah, you go.
You go. You first,
Well,
Neelke.
so we do not often have very viscous samples, or if we have viscous samples, usually after some liquefaction time, the viscosity will be reduced. But we had a few samples where really after 50 minutes there was no reduction in viscosity. And I'm not so fond of using a syringe to break the viscosity, but we put those samples on the device and the recovery was really nice. And it's also mentioned in the leaflet of the device that you can use it for high viscous samples. And so, I mean, the test that we performed was really with the highest viscosity. So recovery is well.
Yeah, and then a question to the two of you, but maybe I'll start with Neelke because you mentioned it in your slide. What about cryopreserved samples? That's a question that's recurring all the time here in the chat.
Yes, so
we have no data, published data. We have some practice, but yeah, where are you with that?
Yeah, so we are currently running a validation on the use of the ZyMot device for cryopreservation in the two ways, as in using the ZyMot device before we cryopreserve, or if we already have a raw sample that was cryopreserved, can we apply the ZyMot device upon thawing? So, but unfortunately, I cannot just share information, but you will hear it when it comes.
And Emma?
Yeah, we only do it the other way around. So we take the semen sample, we do the sperm preparation, and then we freeze. But I would be happy to move into ZyMot for cryopreservation. I think we just need to validate it as you are doing, Neelke, and then we can do the shift because you get such high fraction of progressive motile sperm. So I think it would be beneficial after thawing to not have those.
Yeah. And some people ask, what about the volume? You can then add the volume, some sperm washing media.
Yes.
For the loading of the sample. Yeah, correct.
Yeah.
So I think then epididymal sperm, testicular sperm, not yet there, or can we use it?
Well, we need motile sperm.
Yeah, Neelke, yeah. So question here as well is pentoxypheline. Can we combine that? Do you have experience? I haven't seen any data on this.
No, me neither. And I think it's an intriguing question, honestly. I'm not sure if it will work because usually the sperm, I mean, I don't know if the sperm really starts to become progressive in case it's completely immotile. But I think it's worth a try, absolutely.
Yeah. Someone is asking as well, maybe I can ask that to you, Emma. We sometimes see not a pure normal morphological population, or we even see immotile sperm in that separated fraction. How is that possible?
I don't know because we don't.
And you, Neelke, do you see this?
So the examples that I showed, we had like 97, 98% progressive motility. So it means that yes, 2 to 3% immotile sperm will be visible. I think maybe it's related to the way how you aspirate the final fraction.
Yeah.
But it's
Yeah.
training, of course.
Yeah. Yeah. So one more practical question: bacterial infection, blood cells. Do you have experience with this? How to go about those samples? Emma?
So we do have samples which have elevated levels of white blood cells or red blood cells. Sometimes it has not been a problem using the ZyMOt for those samples. The sperm seems to be perfectly capable of moving past those cells and go through the pores and be collected with high recovery in the outlet port. So that I know. Bacterial contamination, we haven't had any case so far.
Yeah.
So
And you
that's
need to do
promising.
laboratory practice anyway, so you are working in these sterile
Yes.
conditions. So if it is patient related, so you could test that as well beforehand if there is an issue. For IUI, do we really need to take the 500 microliters, but can we take also 300 microliters from the sample? Because it's a large volume to inseminate. Emma?
Oh, okay. Yeah, if you just collect 0.3 ml from the outlet port, you will not get as many sperms. So depending on your threshold or your desired level of progressive sperms to use in an IUI, you would need to validate if it's appropriate for you. Because ideally you would like to avoid centrifugation. And so I understand the question. You would basically just need to run the numbers and see if it's going to work for you, because you will not retrieve as many sperms as if you collect the whole volume.
Yeah. And then I would like to go more, if we still have a few minutes, more on your study, Emma, while Neelke is sorting out her audio. That's okay? Okay, but I'll continue with Emma. So, how do you explain that you have better fertilization rate after the centrifugation? I really want to understand this. The theory behind it, the hypothesis.
Yeah,
Do you have
hypothesis.
Yeah.
The hypothesis provided from the company at the time, which wasn't CooperSurgical, but they were saying that in some cases there's a risk of the fluid that you are retrieving from the outlet port contains seminal plasma factors that are then hindering the sperms to become capacitated enough, or they hinder fertilization. So if you centrifuge, you remove the supernatant and hence you remove the residual seminal plasma factors and you give them fresh media, you will then have a sample without those residual seminal plasma factors. Yeah. That's the scientific explanation that we were given. And we did see some pattern in which patients had this increased risk of total fertilization failure. And it was semen samples that were kind of borderline normal semen parameters. So they had lower concentrations, and maybe they were more diluted, as to say. So I don't know. But with the centrifugation, the total fertilization rate were identical as with density gradient centrifugation. And in those cases, it's something else. It's not the sperm sample itself. It's something in the communication with the egg or the sperm or something like that, which can't be attributed to the sperm prep method itself.
Yeah, okay. That sounds logical. But we could then potentially also do it for IUI, but IUI, we have the sperm need to pass through the uterus, etc., to reach the ovaries, etc. So there is still this dilution out of factors.
Yes.
For you then, Neelke, on your just a scientific question for you around your study, why would you like to analyze the acrosome index if you're doing ICSI?
Yes, indeed. It's because it was a research project at that time with a student on it. And I mean, we had the technique also available. So it was a nice add-on to to really check the techniques and compare the techniques in full. Yeah.
And then you also say that the data set in your study was adjusted for time of abstinence. So could you guide us through what your guidelines are for your patients regarding abstinence? Because we do know the longer we get poorer quality sperms, etc. So what are your current guidelines?
Yeah, the current guidelines are 2 days.
2 days, yeah.
Yes.
And for you, Emma, same?
It's the same. Yeah, we recommend 2 days of abstinence.
Yeah.
Sometimes even shorter if we've had issues in the past with vicious samples or something like that.
Yeah. Okay, lots of questions here. I'm going to take one more, and then unfortunately we will have to close for today. Let me see. Yeah, this is not really ZyMōt related, but yeah, it is related. Sorry. Which device do you then in the end use for IUI, or IVF and ICSI? I want to hear from the both of you. So Emma?
So we use the larger chamber for IVF. We use the smaller chamber for ICSI. Okay. And for IUI, we don't do them right now because we don't do a lot of IUIs.
Yeah. Okay, great.
And then for us, so we use it currently only for ICSI. For ICSI, we use the 850 device. For IVF, I think I could agree with Emma. I think probably both will be okay because for a good sample, honestly, the 850 device gives you sufficient number of total motile sperm count. For IUI, as there is no requirement really to centrifuge before you inseminate, I would be more fond of using the smaller device, the 850, because we will never inseminate 1.5 ml. So in that case, there is no need.
Yeah, perfect. Thank you so much. So let's then conclude. The main messages, I think, from your presentations. It is a sperm separation device. We have 2 types of them. The 850 microliter is 3 ml, and the separation is based on motility. The benefits, easy to use with fewer steps, so that could reduce time, hands-on time, as we learned. It will for sure reduce the risk for errors. It will simplify, but you need to standardize the workflow as well. It gives reproducible and consistent results, and it's clear that the DNA fragmentation is lower after sperm separation, and you will have a higher percentage of motile sperm. There are some reports that you have more euploid embryos, but we need more prospective randomized studies that check all of this in a good and solid way. So I'm inviting you, the audience, to work on this. So thank you very much to our speakers, our expert professionals, for these splendid insights on working with the sperm separation device, ZyMōt. To share, be open about your laboratory and clinical outcomes when you use this device. Also want to thank the audience, the participants out there, and we apologize for the little hiccup in the beginning for joining us. There was a link issue, but I'm sure we are over 180 participants that you were able to reach us. This webinar, as I mentioned before, is recorded, and it will then go onto our Knowledge Hub where you can look at it again should you like to revisit or if you have missed the first part. We do hope you found it interesting and helpful for your lab, clinical work, and you will receive an invitation for your feedback, so please fill it in. Once done, you get a certificate of attendance. If you want to get more information about the product itself, you can indicate that as well in the survey itself. So thank you. And last but not least, I'd like to invite you for our next Expert Insights webinar. We are going to have one in February, and that will be then on the use, the validation, implementation, outcomes, practical aspects, etc. Again, with some great expert users on stage 1 step GM-CSF medium. So we will also invite these 2 speakers again when they have more data on their upcoming project. So that would be great if they want to share it with us. So thank you again, and hope to meet you soon in one of our webinars. Bye-bye.